Good afternoon. I'd like to take you from Singapore to the Red Sea for this project, which is an Israeli, Vietnamese, Italian, and Cypriot project. So we have 4 countries. So I take you to the northern Red Sea in the Gulf of Aqaba, where we talk about Halophila stipulacea. It's the dominant and sometimes only species. It forms extensive meadows along the coast, and we've done a lot of work on mapping. It forms both shallow and deep meadows, as we've seen from Suez. This is 48 meters. For conservation purposes, it's also very important for the region because of ecosystem services to nearby corals, and we're trying to use that for conservation purposes. So what's interesting about Halophila is that it is native to the Red Sea and Indian Ocean, but as soon as the Suez Canal was opened in 1869, very quickly, 25 years later, it became one of the earliest Lessepsian migrants, and it was found here only 25 years after opening the canal, and since then it has spread in most of the Mediterranean Sea, in the eastern part of the Mediterranean Sea. Amazingly, in 2002 it jumped and was found in the Caribbean, and in the last decade, as we've heard from a few lectures here, it has spread in most of the Caribbean nation islands and has even reached Venezuela. Alarming studies in the Caribbean have shown that the species is not very nice, meaning that it is actually quite aggressive and it takes over a local Caribbean native seagrass species. This is quite worrying for the Mediterranean for a few reasons. This is because of the Caribbean work on the invasiveness. This species has made it to this very closed club, this blacklist of the 100 worst invasive Mediterranean species. It's the only seagrass species that has made it to that closed club. Ongoing tropicalization of the Mediterranean means this is real data coming from the East Mediterranean, showing that the Mediterranean has become very warming up and becoming very salty, so it's becoming more and more tropic. And this is causing vast declines in the very endemic and slow-growing Posidonia oceanica. And we've seen a couple of papers showing that this is vast declines in the species. And this is very worrying because the recent expansion, just 3 years ago, of the Suez Canal, meaning that this is going to maybe make everything's going to just go worse. So double trouble maybe in the Mediterranean Sea. So is Halophila stipulacea spreading in the Mediterranean Sea? Well, we have no idea because no one's ever monitored Halophila. It's not been on the radar of most people. Posidonia oceanica and Zosera marina take all their attention away. So the objectives of this work were to try and compare Halophila stipulacea meadows growing in similar habitats for native and invasive populations, comparing morphology, physiology, flowering characteristics, genetic composition of the meadow, associated bacteria composition, and stress resilience using some gene expression, and that will come up later. So how did we do this? We went to Eilat. So this is our Gulf of Aqaba population. This is our native population here in the northern Red Sea. This is how the site looks. These are the temperatures. And then we flew to Cyprus, which is extremely near. It's only a 40-minute flight from Israel. It takes longer to get from me to Tel Aviv than from me to Cyprus. So we went to Cyprus to 2 sites. I'll talk mostly about this site here. And we did this 4 times a year, and we've set up permanent transects in both of these sites here, number 2 and number 1 here. And we followed them 4 times a year every 3 months. And for these sites here, 1 and 3, we also did some flowering work, which co-occurs with the flowering season of 2007, so in July and October. This is how the meadows look like. This is the native meadow, which is only one species. We only have Halophila stipulacea in Eilat. This is how the meadow looks. It's very anthropogenic influences next to these nice Las Vegas hotels here. And the site is in Limassol, in Dream Cafe, one of our invasive sites. It looks very similar. And here, unlike the other site, we have actually 3 species, Halophila, Posidonia oceanica, and Timlosia nodosa. The depth is the same depth, between 3 and 4 meters. So it's a very similar characteristic site. What did we do? We took photoquadrates, as you can see here. We measured a % of cover here, and then we took smaller quadrates where we took morphological characteristics of leaf area, internode distances, number of shoots, percentage of apical shoots, and above and below ground biomass. We also took samples for DNA, bacterial DNA, and we also took some samples for tuberous genotyping, and Kelsey gave a really nice talk about this 2 days ago, so I'm not going to repeat that lecture. Okay, so some results. So in pink we have the native Eilat population, and invasive population is in green. And these are results of the seagrass percentage of cover here. We see that throughout the year there's a much higher percentage of cover in the native population compared to the invasive population. We also see that there's an increase in the summer in the native, while there's a decrease in the summer in Cyprus. Okay, this is biomass above ground biomass here on the left, and on the right below ground biomass. And again, we see that throughout the year also above ground and also below ground biomass is higher in the native population compared to the invasive population. And again, we see that there's an increase in the summer here and also here, while there's a decrease in Cyprus and decrease here in Cyprus in the summer. So native plants increase the biomass during the summertime, and invasive plants are the opposite. They could decrease their biomass through the summertime. So there's a different behavior here. Leaf area, this is square centimeter leaf area throughout the season. We can see that all year round, leaves from Eilat are much larger compared to the ones from Cyprus. And we also see that when you actually scan the leaves and you look at them just visually, they're much more dominant. So you can actually see much larger leaves throughout the year. I think these are the most interesting results. This is internode distances here on the left and percentage of apical shoots. And we see that the plants are morphologically different in Cyprus compared to Eilat. There's many more, the internode distances are larger nearly all year round, and there's many more apical shoots. Percentage of apical shoots is much higher nearly all year round in Cyprus. So morphologically the plants are very different, longer internodes in the invasive and also more apical shoots in the invasive. Okay, these are some sexy pictures from our sexy flowers. These are young male flowers. This is a dioecious plant, so we have male flowers and we have female flowers. These are the pods that were cut, and these are the seeds inside. And what we did is we looked at the percentage of flowers, the percentage of plants that have flowers in blue here. So this is plants in blue. In blue is plants which are either male or female, so percentage of plants that have a flower. And what you can see in these slides is first thing that the season starts quite early. We probably missed the beginning of the season, June, July, but it runs out very early, finishes in September. There's hardly any flowers in September. Compared to, we saw a similar story in one of the sites in Cyprus, but one of the sites in Cyprus, in October we still had 25% of flowers, where plants were still flowering. So we had a much longer season, nearly 2 months longer season compared to the native population. That's very different. Now if I zoom into these blue ones, into the plants that have flowers, and look at sex ratios, so in Israel pink is boy and green is girl. So we have a much more, this is pink is male flowers and green is female flowers. We have a very dominated, female-dominated population in Eilat, and one of the populations that we have here in Cyprus is also female-dominated, but the sex bias ratio in one of the invasive populations was totally male flowers. So we have two major differences. The first difference is that the flowering season is very long in Cyprus compared to a very short season in Israel. And we also have, while Israel it's only female flower, and we've done this, repeated this, I haven't shown you results here, but we have this for a major, this is only one site, we've done this for 6 sites just last week, and it's a similar story. We have only female flowers in Eilat, we have a more plasticity kind of story in Cyprus. Okay, going up to the bacteria now, we've published 2 papers on this recently. The first paper was looking at the same depth but 3 different sites. And we have an anthropogenic influence here, a clean site here and a medium site here. What I show you here is that we have a high microbial diversity in the high nutrients area above ground, and we have a high microbial diversity in the clean site below ground. But what we see here, the most interesting part of these graphs is 2 things. First thing, we see that in every different site, the bacteria is slightly different. So we have some sort of environmental fingerprint which gives us changes in bacteria in different sites. But the most interesting part is actually this tiny circle here, where we have a core microbiome, which is these are shared microbial communities across all 3 sites. So it doesn't matter where you go, you find the same bacteria. Okay, we did another experiment where we did the same site with 4 different depths, 4, 9, 18, and 28 meters in the clean site, and we have here the same story. Yes, bacteria change in every one of the 4 depths, above ground and below ground, but they also have a core microbiome where the bacteria are shared throughout every single depth. There's a group of bacteria that are always sticking to the seagrass wherever we go. So these are really preliminary results. They were just analyzed in a very crude way. In the meantime, we're still working on this, but this is from the South Beach, the same site I just showed you now. And this is from Dream Cafe in Cyprus. And I'm not going to go into these results, but what we see here, first thing is a much lower diversity in the bacteria in the Cyprus plant. So we're still working on this. It's very fresh from. So to try and summarize, in the meantime, Cyprus invasive and native populations from Eilat are quite morphologically different. The invasive plants are smaller, they have more apical shoots and the larger internode distances. If you think about invasive plants, maybe these morphological characteristics alongside the plasticity of the sex ratio, which seems to be much more plastic in Cyprus, could give an advantage to an invasive population. Maybe this is helping to fill up these empty spaces due to the disappearance of local seagrasses like Poseidon in the Mediterranean. So this might be an alarming news. And the epiphytic community, we're still working on this. It shows we've done more work that I haven't shown, showing that it's reduced diversity, but we don't know exactly the function of these bacteria. We're still working on this right now. And the most interesting part is, so the invasiveness of Halophila has been attributed to its ability to adapt to a very wide range of conditions. Different salinities, different light levels, different nutrients, and maybe different temperatures. And the main question we had was, are there differences in the resilience, thermal tolerance of the invasive and native populations? And here I give a commercial to Hung's lecture tomorrow, where we actually flew to Cyprus and brought plants back to Israel and grew them in the mesocosm, did some nice work, including some nice gene expression work. And lastly, I would finish by saying that we need much more Halophila mapping and monitoring needed, because it has not been on the attention of anyone really to monitor Halophila in the Mediterranean Sea. And this is for Peter Ralph. I'm not sure, yeah, he's still here. So we've just come up with some work combining physiology, transcriptomics, and metabolomic profiling approaches in Halophila. So just wait, and we even have that which just got accepted last night, and the genome is being sequenced as we speak right now in Illinois in the States. So that's going to come up and help to close some of the gaps. And I'd like to thank my funding, and students are always welcome.
Thank you, Gidon. That's a lot of information. Any questions from the floor?
Hi. Gidon, you found differences in the morphology between Cyprus and Eilat, right? Can this be explained by differences between sexes? Because
between sexes?
Between sexes, between male and female plants, because you have mostly what is male plants in Cyprus and female in Eilat. Yeah. Are there differences between the males?
But the differences were all year round, and we
like
only found sexual reproduction only in the summer. So sorry? Even the differences in leaf area were there all year round. Here. So all year round, sorry, this graph here. All year round, you have leaf area is much larger, not only in the summer where there's reproduction, sexual reproduction. So I don't know if a plant is a male or female if I come in the winter, but even then the leaf area is larger compared to Cyprus. Not only if it's a male, not only when it's flowering.
So, okay, so you don't know, you only know if it's a male or female when it's flowering, so you don't know if the population is dominated by females or males.
What Yossi Loya has done on corals, he's put a sign on a coral and he comes back to the same coral and see if it changes between males and females. Yes. It could be related to light. We haven't measured light. It is the same depth, but it doesn't mean it's the same light. It could be light. Yeah, but I haven't got money to buy light sensors.
Well, thanks. I was just wondering to which extent you can really explain your differences between like native versus invasive plants, and if it's not really like a location effect that you're looking at, because you only looked at two sides. Would you expect to find the same results all over, for instance, the Red Sea and the Mediterranean?
So in the Red Sea, but which part of the results? So the flowering, we've extended I haven't shown this, but for the Red Sea we've done at least 6 sites already, not just one site, and the flowering story is exactly the same like the one site. I haven't been able to do this in Cyprus because we haven't been able to find many more relevant similar sites. It's not fair to find a deeper site, or it has to be the same, otherwise I'm comparing apples and oranges. So it's not so easy to find a mixed meadow in the same depth with Halophila and other, so I hope, but we have also brought the plants to the lab, and that's tomorrow's lecture, and they have exactly the same conditions, same sediment, same aquarium, everything's the same light, exactly the same, and then they behave totally different.
Thank you for the talk, Gidon. I really enjoyed it. I had a question about the different sites as well. The non-native site seemed to have a lower cover than your natural site, and I just, based on those pictures, maybe your data wouldn't show that, but when you have like quite sparse patches, you usually find much longer internodes and more branching. So I just thought maybe that could be another explanation, that it's just actually the cover or the space at the site that might be affecting that trait.
I totally agree. But again, if you bring them into the lab, into the mesocosm, same conditions, solve that problem.
Yeah. Yeah, that would be great. And the other question I had in relation to your flowering and your sex ratio is where you only find females, do they develop seeds?
Yes, they do. I showed pictures of pods and seeds.
Yeah, but how do they
No, there are
make
males. I
seeds
showed there was a small amount of males in there. I showed their pink. Okay, here. So we have males. We can't have seeds without males. So there is some pink males in the population, but they disappear really fast. While the females, you could find them for 3-4 months afterwards.
Right. So
Here, yeah.
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 Great. So it's maybe a timing thing that the males might come out a bit earlier.
Yeah.
Yeah. Great. Thank you.
I didn't mention this. We think it's maybe related to the investment in for an invasive population, the investment in females is quite large. That's the theory. So maybe it's easy to have quick males and just survive with males. I would also mention this is the first time that females and males have been found in the Mediterranean, and the theory has been so far that the water's been too cold, either that the water's been too cold for the development of female populations, or that the first clonal plant which arrived, the fragment which arrived and started the whole story in the Mediterranean was a male flower, because so far Procaccini and Gambia have only found male flowers, and this is quite new. This story is a big story.
Getting a quick question. Looking forward to seeing the manuscript on the transcripts, but with your core microbiome, did you get data on the archaea and fungi as well? Or you
On the
only
archaea
just
and
and the fungi?
No.
No,
Archaea,
so-
yes, fungi not.
Okay.
Yeah.
But-
We've done some microscopy on the fungi.
Okay.
Yeah.
Okay, because it could be interesting. There could be a core fungal
Yeah.
component that's also going through it.
Yeah. I haven't shown all the micro. That wasn't the topic of the lecture.
Yeah. No worries. Next time.